All right, great. So everybody, welcome to today's MPRINT webinar series. My name is Prince Cannon Carroll. I'm the PI at the Vanderbilt site, and today I'm pleased to introduce one of our own. This is Wei-Qi Wei. Wei-Qi is Associate Professor of Biomedical Informatics here at Vanderbilt University, and he is the PI for our phenotyping core. And so as part of that, and as part of some related projects, Wei-Qi is an expert using informatics tools, including machine learning, natural language processing, and so on, to harvest knowledge using big data sources and genetic data sources. So he's involved in multiple large networks and collaborations. He's trained a number of investigators and published over 200 research papers. Before I have him start, I'll just remind everyone the chat is enabled, so you can ask questions in the chat. You can also use them in the Q&A, and I'll be looking out for them, and we'll probably have Wei-Qi answer questions at the end. With that, Wei-Qi, you can go ahead.
Sure. Thanks, Prince, for the introduction. So, hello, everyone. So, sorry, excuse me. So I guess this talk, my intention is to, split the talk into two sections. So the first half, we're going to talk about the EHR and the limitation of using EHR for your research and what lessons we learned so far, after, using EHR for research practice for over 2 decades. And the second part, I'm going to show you some research examples because it's a, pediatric, pharmacogenomic, seminar. So probably going to focus on, some research from our lab, related to medications. So EHR research has been, growing rapidly over the past 2 decades. Has been widely recognized that EHR is it's a major and invaluable, resource for conducting, biomedical research. So as shown, from this figure, you can see, from the year 2000 to last year, you can see the number of publications based on the PubMed. You can see the number is continuously growing, and it's pretty fast. And so right now we're looking for like 10,000 unique journal papers every year talking about using EHR for the research. And you can see the number is actually doubled every 4 years. And the orange section here shows, represent the VUMC, as a single institute contribution. You can see that, as a single institute, we make a contribution each year, as the probably about like a 2 to 3% of the, total EHR research in terms of a publication. So in addition, phenotyping, or the EHR research has become some, the backbone of several large national consortiums, collaborations. For example, like eMERGE network. We're going to talk about some work from eMERGE network. All of us, you already heard that, and of course, MPRINT. All right. So just give you rough idea about the number regarding EHR. This is from a single medical center. This is Vanderbilt data. Just show you the number here at Vanderbilt. So Vanderbilt EHR system has a huge amount of healthcare data. So we have over 2 decades, at least 2 decades, data accumulated in our system. So every year, the system, we capture like 10 million prescription information and 100 million lab test results and 600,000 new diagnoses and 4 million procedures and 15 million clinical notes as well, So this database, so far we have like maintained information over like approximate 4 million unique individuals. And among those 4 million individuals, we have 300,000 have their DNA linked with their EHR. And we have another like 100,000 who already been genotyped, and another 100,000 has already been sequenced. And this number, remember, this number are going to be, keep growing and make it, Vanderbilt biobank is the largest single institute biobank in the world. So in order to take advantage of, the huge amount of EHR data, like Vanderbilt data, for clinical research. Phenotyping is a basic step. So basically, phenotyping means phenotyping is by definition is talking about the developing, standardizing, implementing and organizing algorithm or your strategy or your tools and knowledge base to accurately help you to identify individuals' phenotype status from clinical data. So the quality of phenotyping is super important to your following studies. So simply say that if your phenotype, algorithm is not accurate, your research conclusion may not be, accurate as well. So, when I started doing phenotyping, I thought it would be a super straightforward task. I thought, probably just, find the diagnosis of that patient, and then probably the job is done. However, I quickly realized probably it's how naive, that notion was. So there are so many, big challenges in using EHR data for phenotyping. I'm gonna list here some major ones. We're gonna talk about that one by one. Chart fragmentation, inaccuracy of EHR data, different challenges to pull the data out, terminology gaps, etc., etc. So just give you a rough idea, on the right side actually is a real example of EHR-based algorithm. Actually, this is created by the eMERGE network, and this is the very first EHR-based algorithm has been published. That's published around 2009 or 2010, around that time window. So you can see, in order to get a good quality, phenotyping results, a good algorithm requires the combination of multiple data elements from EHR, including differential diagnosis, different related lab test results, medications, and clinical notes. So it's a far more complex than it initially appears. So first thing I'm going to talk about the challenge of using EHR is a chart fragmentation. You consider there are different type of chart fragmentation issue. One clear one is like patient went to a different hospital, went to see different health care providers. That's one. You don't have a complete medical records, and the different EHR system, they don't talk to each other. Therefore, you only have a partial information of that patient. So that's one chart fragmentation issue. The other one is, remember, although we already have right now EHR is everywhere, and at Vanderbilt we have like 2 decades of EHR data, but clearly there's some loss of information. For example, historical information, before year 2010 or year before year 2000, that's a paper based, and you want to be able to get those information. And it's not trivial task to transfer all those paperwork to digitalized version. Is basically impossible. So that's another type of information. We call it longitudinal. You have a short-term EHR information versus you have a complete long-term information. All right. So the early study from this lab is about 10 years ago when we started doing this is the first idea we're going to be, say we have one single medical center versus, say, I have a relatively complete medical records. We want to see the how it affect the phenotyping performance. So for the single medical center data, we use the Mayo clinical data. And so from Rochester, Mayo clinical data. So that's a single medical center data. And in order to get a relatively complete medical record information, we use the Mayo clinical data plus the OMC. OMC standing for Olmsted County Medical Center. So basically, there's a very unique resource back to that time is in Rochester, Minnesota, they have an epidemiology project called Rochester Epidemiology Project. So basically in that project, they link all the information, patient's information, and including Mayo Clinic and all the, Olmsted County Hospital information together. So therefore, you have, suppose you have a relatively complete medical records, because if people familiar with Rochester, Minnesota, that's a relatively isolated from some other big cities. You need to drive like at least 1.5 hours to the closest big city, which is Twin City, And also, majority of Olmsted County Hospital residents, they're living in the, the neighborhood. So in that case, we run the run a phenotyping algorithm. We compare the performance using only the data from Mayo Clinic versus we assume that's the complete medical records. And when you see the results, you can see that if you look at the PPV here, which standing for positive predictive value, which means you identify the patient, say this is a case, and how many of them percentage are true cases based on meta review. So you can see the performance didn't drop a lot. So which means if you design a very nice algorithm, and adding more information probably won't hurt you too much because you already got a sufficient evidence to prove that patient is a case. All right. The major problem is this. The major problem is the sensitivity or the recall, which means if you don't have complete medical records or you don't have lots of information, you're probably gonna lose, a big number of cases. All right, that's the big lesson we learned. And another thing we learned is that we talk about that is you have a short-term medical record information versus you have relatively long medical record information. So back to that time, we used over 10 years medical records as a gold standard. We compare that, and the conclusion is like if you are looking phenotyping a common disease, you need at least in order to get a reasonable good performance, you need at least 5 years of medical records. All right. So another second challenge I want to talk about using EHR is the inaccuracy. As inaccuracy is a big challenge. Some people may argue, like diagnosis should be accurate. But let me give you some examples. HIV is a good example. There are some patients, has only one HIV code. And when we doing the meta-chart review, we noticed that actually this is, it's a cut and a contact with another patient who got the HIV tested positive. So in that case, the patient, immediately, put on the, antivirus medication and send the blood test to the lab, but eventually turned out to be negative. So, single one code is not accurate enough. There's many other examples, cancer code is another great example. Lots of patients with one single cancer code, usually just a biopsy, requirement of biopsy. And IMI is a heart attack is another great example. Ear doctor signed an IMI code there, but a simple one, diagnosis sometimes is not accurate. It may rule out due to the lab test results or others. All right, so this figure on the left shows the distribution of patients with only ICD codes, with only medications, relevant medications, or with only the phenotype document in their problem list. And of course, some patients with overlap. But you can see there's a large number of patients have only diagnosis without any supporting information. That crossed, many common disease. We just show 4 common disease here. Actually, in the original study, we screened like 10 different diseases. We just randomly pick 4 to show you here. Breast cancer, HIV, rheumatoid arthritis, type 2 diabetes. So a big number of patients, they only have diagnosis. They don't have any support information. So, which means, raised a major concern about their true, phenotype status. So the right figure shows, from our study that when you combine information from, using multiple EHR elements, for example, you combine diagnosis plus the relevant medications to confirm a case. You can see, or you can, it's a problem list plus the medication. And you can see when you use at least 2 or more components, EHR elements together, confirm that, you can see the performance will significantly increase. So, right. So the third one I'm going to talk about the using EHR data is that, not all EHR data can be extracted equally. From the left to right, you can see the left is the structured information. Example is like diagnosis, lab test results, prescription database, and procedure information. Those kind of information are super straightforward to do. Run a SQL, you can get those information very easy. And the middle part, I think, is I always say middle part is like for example, it's like we call that semi-structured information. It's like a patient may have like a problem list, medication list, allergy list, and we're gonna talk about allergy list later. So I think those information is they have a semi-structured and those information are relatively easy to put out. You don't need to write a super complicated NLP, natural language processing pipeline to do that. You can easily using regex, regular expression to pull out this information accurately, and those information is super valuable. And I think, in my personal opinion, it's like the semi-structured information in the HR has been underused. I think it's a super valuable. Of course, on the right side is unstructured information, radiology report, admission notes, progress report, and discharge notes, etc., etc. They have lots of information in notes. And many symptoms, but the problem with that is that, that's a free text and it can be described in so many different ways. So it's notoriously messy to, put out information, and different institutes may have a different way, to do that. So you have to, so in order to get those information, you may have to, go to some complicated natural language processing pipeline to do. So here's the lessons. We are not going to go to the details, or teach you how to do the NLP in this talk. So I'm just, and personal, in my personal opinion, I think that NLP will pretty soon, being replaced by large language model, my personal opinion. But I think I want to share some, some lessons we learned from the last two, decades of using NLP. And I think, most of them you can apply to AI or large language model in the future as well. And so first one is, NLP require, extra privacy protection. It seems like you don't want to directly get the patient notes into input into, the ChatGPT, something like that. So privacy is a concern. And also there's a technical, concern. You have to have a special server set up in order to, make the pipeline running. It's similar for AI, okay? And another one is, the second one is compared to traditional or common phenotyping algorithm based on SQL, NLP software has some, may have some, IP or intellectual property issue. The reason is like a lot of NLP pipeline for AI as well. You're probably gonna have some, there's a part of the institute's, decision-making part components. So therefore, the code may have some intellectual property issue. When you share the code with others, they'll probably, have additional barrier, in order to share the information, share the code with others. And third one is like NLP performance, may vary across different sites because of heterogeneous issue too. And it's I guess this observation also apply to machine learning algorithm. You probably heard that lots of machine learning algorithms. When you train at your local performance, local data, and test that the performance looks nice, but when you generalize the model to other place, usually the performance is gonna drop. All right. And the negation, we don't want to go to the negation. Negation is still challenges, for NLP problem. And the last one I want to particularly talk about, is NLP pipeline may not be generalized to rare phenotypes. I want to say that is, it applies to AI as well. I think the most important thing is that, when you do a research, you have to make sure you have the data. All right, like we have a huge amount of EHR, but doesn't guarantee we have enough rare disease information with our system. If you don't have those information, advanced technology, whatever NLP, artificial intelligence, or whatever the fancy, chat box available won't be able to help you. You don't have the information, you don't have the information. All right. So next challenge, unique challenge from EHR using EHR is the I call the gap between different terminologies. This is very unique to EHR system. So consider this, the three components of clinical practice, diagnosis, and when you get the diagnosis, you get the treatment. Okay, give the patient treatment. Then the lab test and the symptom gonna help you to confirm the diagnosis and also confirm the treatment. Okay, so logically, the three components link to each other. And example here is like type 2 diabetes. The code is we here is diagnosis code is 250.1 250.0, 250.2, or E11, that's ICD-10 and ICD-9 codes. All right, the green bar, the code is basically is RX norm codes. Each represent a concept of a unique metformin, different dose or different form. Similar for the right blue bar, here shows the hemoglobin A1C. The problem is this. The problem is that you have a three linked components: diagnosis, um, medication or treatment, and lab test. And each clinical domain, they have their own dominant terminology. So for example, here is like in diagnosis, people commonly use ICD codes. All right, for medications, people commonly use RX norm, and procedures, most people use the CPT codes. And similar for lab test results, line code is designed for represent the lab information within EHR. So the problem is, those terminologies, they don't talk to each other. Okay, for example, you have the code here, diagnosis code here, those ICD codes here. By a computer, they don't automatically understand, how 6800, which is a metformin represent for metformin in RxQE, how they link to 250.0, 250.2 in diagnosis. So that's a big problem. So 10 years ago, we start, tackle this problem. This is a huge problem in informatics. So what we decided to go, we're gonna go with the super straightforward, simple relationship. We only, talk about, for example, indication. So we want to start with the link, RX norm concepts with the ICD codes, just based on, indication relationship. So what we go is like, what we did is that we start RxQE and we go through, multiple resources, reliable resource, RxNorm, Cider, MyLab Plus, Wikipedia. Of course, it's a 2-hour, free text. We have to go through NLP process to get the disease they mentioned in their description about the medication. Then we just vote. The idea basically is if 2 or more resources mentioned, this is an indication. We assume that's reliable. And based on meta-chart review, we deliver, a resource. We call that MEDI, basically link the particular RxQE to their indications. So let me give you an example. As another, I think it's a very important thing for that resource is like we calculate the prevalence for each indication. So that means it's prevalence is defined by the unique number of patients with both medication and the indication in record, divided by a patient with using that medication. Okay, so why it's useful? Consider this. This is RxQE for metformin. You can see, based on the MEDI, they have multiple indications. Type 2 diabetes, obesity, and PCOS. But you can see, based on the prevalence, you can easily tell to say, given a patient with metformin, there's 80% chance that patient got diagnosed with the type 2 diabetes, and a very small chance a patient with the PCOS. So people use that resource they can do phenotyping, they can do, adverse drug reaction, research. And so MEDI was pretty successful. Lots of people using MEDI. So, excuse me. So we move on. This time we call that FMAP. What we gonna do is, we're gonna link all the biomedical or clinical terminology together. So we use a very similar approach, and let me just show you, the results. It's a 2 similar disease, like type 1 diabetes, type 2 diabetes. You can see based on the concepts, as for type 1 diabetes, the most important concepts is, diagnosis, and you can see the findings are very different because the finding in type 1 diabetes, there's a very strong, indicator is if their onset age is much younger. And you can see the treatment is very different. Similar, for type 2 diabetes, other than diagnosis, you can see, here's a relevant test, and this is, the first-line treatment for type 2 diabetes. So this is the phenotyping is costs a lot. It's very expensive. And this chart just to show you what is the current process to develop a phenotyping algorithm. Okay, so we usually start with a person who got an expert in in those domain, in that phenotyping, in that disease domain. So that person usually, create an algorithm, draft an algorithm. Then we have an informatics guy help that people to convert you or translate the draft into a coded algorithm, then implement the algorithm against the or EHR database. Then we just did a manual chart, first run a manual chart review and see how the performance looks like. If the performance is not good enough, we're going to modify, go back to the domain expert, modify the algorithm, rerun that again, reevaluate it. Usually the process is going to go multiple runs until we say, the performance looks good. We're going to finalize the algorithm and share the algorithm with others. So there's I just want to say, there's a two major, issue with that. Number one is to consider this is that usually we start with the domain expert. Right now we have some AI tools available. So if we can leverage AI tools to help us do this, they're going to save us lots of time. So in that case, the physician or the domain expert is not create an algorithm from scratch. They're gonna just review an algorithm and make some suggestion. They're gonna save lots of time. And another important thing I think is that we need to mention, emphasize is, in the last 10 years, we developed lots of algorithms, but the problem usually is like once the algorithm has been released to public audience, usually that's marked as the end of the journey, and nobody gonna very few people are gonna be reuse the algorithm and upgrade the algorithm, which will represent a huge loss for phenotyping field. So here's let me show you some numbers to show the phenotyping challenges. So this slide just highlights the number of phenotyping algorithms developed by the eMERGE network over a 5-year period. So for those people who are not familiar with the eMERGE network, it's a NIH-sponsored national consortium focused on using EHR data for genomic medicine. So the network, eMERGE network, includes 11 leading medical centers such as Vanderbilt, Harvard, Columbia, Mayo Clinic, UAB, etc. So those people are, the network are very familiar with the EHR. But even though, it took us like 5 years and the contribution from experts across 11 medical centers to create a, to generate just 25 algorithms, you can see from the number here. And based on the lessons, based on the the experience with the eMERGE network. Sorry, can you see my screen?
I just went back to your, it looks like the screensaver instead of the slides.
Yeah, it's weird. My PPT just stopped. Give me a second. Let me quickly restart it.
Sure.
So where are we? Can you see that right now?
Yeah, that looks like your first slide there.
Right. Let me go to here.
Yep, perfect. Okay.
Perfect. Okay. So yeah. So I guess two lessons we learned from the eMERGE experience regarding phenotyping part is number one is like it's super expensive and like what I said, it took a long time to get a final product. So if you think about it, if you have like your institute, you have like hundreds different researchers, and each one had their own target phenotype. If you go to the traditional way to develop an algorithm for each of them, it's basically impossible. That's a super time consuming. And another lessons we learned, like what I said, is like human domain expert really can deliver or generate a very high quality algorithm. Like you see here, the PPV, which means that the accuracy basically is 0.95, that's a super high, accurate algorithm. If we don't reuse the algorithm, we're gonna waste waste the resource we just generated. Okay, so some people say, if developing algorithms is super expensive, why not, we go leverage the power of a computer to do some, high throughput phenotyping, stuff. That's the hot topic. We want to go too much detail there. Just show you example. People may heard the word like a fee code. A fee code is basically group a similar diagnosis together. So in that case, a group similar diagnosis together as a phenotype that's going to be used as a bridge for a true phenotype status. So majority of the people are using right now is created from my lab is by my student Patrick Wu. That's the ICD-10 version. You can see the panel A and the panel B basically shows the coverage of the ICD-10-CM. We cover lots of ICD-10-CM, and as well as the international version of the international version of ICD-10. And some code we missed. Usually that's just local codes. That's just not commonly used. And of course, this is a demo, just a demonstration to show that the performance between the old version and the new version. I mean, old version is the ICD-9 version and the ICD-10 version. They're very similar. And so beyond the fee code, is the because it's a pediatric, audience. So this is a work we recently done, this year. It's by MD PhD student from my lab, Monica. So the idea basically, we want to create a special fee code for pediatric or little patient. So there's many reasons doing that. Number one is that, pediatric patients, they have a different disease spectrum. That's very different from adults. And but the traditional fee code was purely based on informatics way to manual review that based on the ICD diagnosis, which is a population based. The majority are adults. So therefore, we want to create a new version based on the fee code. We want to create a pediatric version. So we have we apply a hybrid approach. For that hybrid approach is with bottom up and the top down. So bottom up is that we want to identify existing fee code that are rarely used in pediatric patient population. Then we review them, and we're going to decide whether or not they should be removed, or we just keep those because they are represent for rare disease. Okay, and for the top-down approach here is like we focus on fee codes with a pretty higher prevalence amount of or pediatric patients, and then we consider if we want to add some new children codes for those codes with lots of patients. And quickly show you the comparison of the pediatric fee code and original fee code. You can see we add about 358 new fee codes. That's a special design for pediatric patients. And we remove, a large number of almost like a 200 original fee codes. That's probably some lung cancer, etc., and probably like a dementia, etc. So this is a comparison. We applied the pediatric fee code and validated the pediatric fee code against the GWAS catalog. We used all the major findings, report findings from the GWAS catalog, for a pediatric, a cohort. And you can see, the replication looks very nice. And the pediatric, of course, is much better than original fee code. And of course, the pediatric fee code give the much better p-values and give you a better p-value. Give you a better statistical power to do your research. So that's the 30 minutes about the informatics part. Now let me move on to show you some examples. So the examples, here are the data, show you the data. Top are EHR data, and lower are the linked DNA information. Once you got this information, you can do different type of researchers. Different type of research studies. You can do genome-wide association study, traditional. You can go up to do some pheno-wide association study given different genetic difference. And of course, you can leverage EHR to do lots of observation study. If I got time, I'm probably going to talk about the AI study as well. So let me start with the statin. Statin is the most common medication, I guess, probably not #1 medication in the world. And so basically, statin reduced the low density of lipoprotein or bad cholesterol, LDL, in plasma. So the reason is LDL is highly associated with the cardiovascular disease. So statin has been widely used in that case to prevent cardiovascular disease. So there are probably around like 30% of adults over 40 years old in the US right now are taking statins. and the number is increasing as well. The reason is like, scientific evidence to show the benefits of early prevention for cardiovascular disease, and also shows the benefits of, lowering bad cholesterol, right? So, in fact, studying a very, very, effective, in LDL reduction, previous research shows that every 1 unit reduction in LDL lower probably about 20 to 30% of CVD instance. And however, even, patients under statin treatment, we still observe lots of patients develop a CVD, but others not. So we call that, in that case, we call that residual CVD, okay? So the two questions we try to answer in the past few years relate to that. So number one is, are there any genetic variants beyond LDL that may increase the risk of residual CVD? Okay, the second one is because we observe, each individual may have a very different response to statin, and even under the same dose. Okay, so in that case, we try to understand if the individual response difference will be, are they independent factor associated with the residual CVD? So that's the two questions we try to answer. So in order to do the first one, come back to the first study, if there are any genetic variants that's independently associated with the CVD, beyond LDL. So what we design is a case and control study, okay? We need to develop a phenotyping algorithm to identify patients taking statin and developing CVD, that's our case. And also, create a cohort of a control are those patients taking statin, but they don't have a CVD, okay? So this is the algorithm, for how to identify cases. So for this study, we have a statin mentioned at the beginning, and we have a buffer window to make sure the statin is not prescribed due to CVD event. So therefore we have a 7 days buffer time there. And after that, a patient may have a, CVD. We define CVD as, either MI or the patient requires some, cardiac procedures, immediate procedures. And we talk about that MI is not heart attack. It's not very easy to identify. So we design an algorithm to do that, which means, by definition, is we require within a 5-day time window, we require multiple MI code, and it's a plus a confirmed lab. It's a troponin or CKMB ratio. The algorithm looks pretty nice. We achieved the performance like 95 or 98, regarding the PPV, positive predictive value, and in the validation set, and looks, very similar performance. So we move on and run a genome-wide association study. So for people who are not familiar with the Manhattan plot, is the lower number here, X, shows the 21 chromosome. And the Y here, the basically is the p-value, the smaller the better. And you can see probably one little, it's either pink or red, line here. That means the Bonferroni line, which means if anything beyond that line is not a random by chance. It's potentially it's a statistically important signal, and the signal here basically is it's coming from a gene called LPA, and the top SNP here, 1 or 2 here, actually that 2 small SNPs explain that about around like 30% of variance in Lp(a) levels in plasma. Okay, of course, we run lots of, validation studies. We have additional cohort supported by Riken, Vanderbilt cohort as an independent one. We also, run the GWAS in Geisinger and Mayo as well. Here's the meta-analysis. So, and we also conduct, survival analysis for the signal. We have 3 groups based on the copy number of genetic variants. The top one is just a 0 copy of that risk allele. And the middle one here is the, risk allele, just 1 copy of that risk allele. And the lower one is the, those patients with the 2 copy alleles, risk alleles. You can see clearly there's a big difference, among the groups. And we also adjust for different things. At that time, we also find a different replication cohort from Harvard. And then we adjust for, the LDL-C. We also adjust for the, the LDL-C change, the bad cholesterol change as well. And we also, I think the very interesting thing is here is the LDL-C lower than 70, milligram per deciliter group. So we're doing that. The reason is like, for those groups, there are patients taking statin, their LDL is super low. It's great. So we're trying to see, is it additional reason that, for those patients, once their LDL is good enough, should we, still have the risk, for the residual CVD? All right, you can see that's confirmed that. That's great. So what lessons we learned? So the results we learned is, yes, we find an independent one. LDL, LPA as an independent, genetic variants that make an independent contribution to residual CVD. But for us, the question is like, if we design a medication to target, LPA, what's gonna happen, right? It's like, is there patient going to increase the risk for other disease phenotypes? For example, you don't want the patient lower the LPA, but increase the risk for, type 2 diabetes, etc. So we can use a pheno-wide association study, or PheWAS, to address that question. Remember, the top signals we show is like the top two, SNPs that explain like a 30% of the LP-A level in plasma. So we put the patient, as a case and control groups based on that SNP status, and we run a pheno-wide association study. And you can see, the first round you can see is that LPA is only associated with either hyperlipidemia or cardiovascular disease. And when we adjust for LPA, when we adjust for LDL, you can see the signal is gone, which means of course it's confirmed more findings say that's an independent factor to cardiovascular disease. But also it suggests that if we target LPA as a drug target, it won't hurt us. It only bring the benefits for cardiovascular events, reduced cardiovascular events. All right, let's move on, talk about individual response to study. So why is important? I think it's a very unique study, we can do using EHR. Consider this if we compare this to a trial. In trial, you have, for example, you have two patients, patient A and patient B. And if you assign like a 10 milligram to those patients, you're probably going to see, you're going to classify these two patients together. But actually, if you look at their drug response, medication dose response, you can see the two patients are very different. Okay. So I guess, in order to describe each individual's, medication or drug response, you need three parameters. One is E0. E0 means like a baseline LDL in this case, which means is you don't take medication. So what is the lipid LDL looks like? Okay, that's a baseline LDL. Another one is the Emax. Emax means is that you can keep increasing the dose for LDL theoretically, and but the cholesterol won't go any lower. That's the maximum reduction of the LDL called Emax, right? And third one is the ED50, or some people call that potency, which means is like what is the dose for that individual to reach half Emax? Okay, if you got the three parameters, you can draw the line, the individual, drug repurposing line. Drug response line. And so, but in order to do that, it's quite complicated. The reason is like, you have to have a patients with the baseline LDL, multiple doses mentioned in their medical records, and multiple LDL mentioned during the different time window. So let me quickly show you this. The number, remember we have 4 million individuals. So after we apply those rules, you can see, remember statin is the most common medications. You can see we only can get probably 1,600 atorvastatin patients and 1,000 simvastatin and just a few hundred rovastatins. And of course, the demographic shows it's very similar. It's no big difference and a very, a reasonable demographic data. And here the panel A basically show the raw data. And you can see from the rosuvastatin here, you can see pretty much makes sense. When the dose increase, you can see the LDL just reduced. And the middle column shows the distribution of Emax, basically shows, like a atorvastatin and a rosuvastatin actually has more efficacious than simvastatin. And the right part shows the potency, individual potency. You can see each individual may have a very different, potency. And clearly, and the resource study potency is better, is higher than a TORVA study. A TORVA is better than sigma study. So we put the individual factors E0 and Emax and the potency, each individual's potency into the model, and you can see that's the clearly along with other covariates like total LDL reduction dose, prior CVD events. Of course, if the patient got a prior risk, that's going to be a big contribution, to the future CVD events. But you can see here is like both Emax and ED50 are very independently associated with the risk. Higher Emax people have higher risk of developing CVD, and potentially means they have a larger a place to decrease their LDL. And we're trying to see if we can replicate the ED50 the signal in a different cohort. Okay, so that's the starting story. Like what I said, in the HR, we have lots of, semi-structured elements. I think we just understudied, underused. I think that for this particular, pharmacogenomic space, I think, I want to focus on, on the allergy box, allergy section. Allergy is, because for the pharmacogenomic research, it's often the challenge is the smaller sample size. So in this study, I'm to show here, I call that allergy was. So what we did is we put the information, available information from the allergy box. We got everybody there, and we're checking their drug allergy label and to see what is the case going to be patient taking that medication and document that medication as allergy in their allergy box. And control going to be patient taking the medication, but they don't have those medication document in their allergy box. You can see the top one medications, lots of patients mention penicillin, in their allergy box. Then the next one going to be sulfa and codeine, et cetera. So we run a genome-wide association study for each one of them, and you can see just two interesting signals. We have a bunch of signals. I think two interesting one. First one is like the codeine and the CYP2D6. That one has also been independently, replicated. I think it's in the independent group is, I think they are using 23andMe. And another interesting study, interesting finding is the penicillin and HLA region. So around that time we published this study, and there was a European group approaching us to say they have the exactly the same signal as well. So we combine our data together, and that work has been published on AJHG. Quickly talk about some other ideas of using drug label. So this is we call like a drug-to-drug interaction wide association study. So the idea basically is we're looking for, we're going to scan the medical records in their allergy box, and we're looking for, the most frequently documented medication pairs in allergy box that potentially going to help us to, identify some unknown drug-to-drug interactions. So in the preliminary data, in the pilot study, when you apply this approach, so we used a TORVA study as an example. We have looking for EHR database, looking for patient taking a TORVA study. Then we have a patient who are case going to be taking a TORVA study, but they also have a TORVA study document in their allergy box. And control going to be patient taking a TORVA study, but they don't have a they don't have a TORVA study or Lipitor document in their allergy box. So for case, we just look back to say this is the first time they have a TORVA study in their allergy box. We look back to 6 months, see what medication they took around that time. And for controls, we use the data is like what's the last time they in their EHR their final prescription date for a TORVA study. We look back 6 months. Then we just compare the medications, and you can see from the volcano plot, it's like we replicate, a lots of already known drug-to-drug interaction. Also, we identify some new ones. So this is a feature article last year from Nature Communication from this group. So the idea is like we want to create a high throughput approach using multi-omic data. using multi-omic data to do a drug repurposing research. So idea basically is like we are quite familiar with the EHR data, but the problem is like if we using EHR data to do drug repurposing mining, we're gonna lose our resource to do a validation. So we decide to generate the hypothesis from another place. So what we did is this. So we're gonna start with the genome-wide association study. We used the summary statistics and the predictive scan to get their gene expression, estimate the gene expression up and down. Then we just go to the drug database and looking for, are there any existing drug can switch, can just reverse the gene expression. So then once we got the medication list, we're gonna go to the EHR system, and of course we're gonna validate the signal as well from all of us. So in the pilot study, we test two, we find it probably like in total like 30 medications for hyperlipidemia and hypertension, and we have like 20 of them already in the practice use, and we have a 10 interesting findings about that. So let me quickly talk about the large language model. So what is a large language model? So basically, large language model is like it's a model that trained by huge amount of text and map the relationship of different words onto a huge amount of, you can consider as a neural network up there. Okay. So if you project that network in a 2D dimension, you're probably going to see, something like this, here the men and women, they have a similar location like a king and a queen. So in the context, say they talk about the man and the king and the women, you probably refer, you can defer that, when they talk about the women in that context, that refers to, a queen. Okay. That's a large language model. So people heard lots about, the most popular application of a large language model is ChatGPT. So what is ChatGPT is doing? Basically ChatGPT is like your input is a sequence of a series of words. You give them a sequence of words. Then the ChatGPT gonna predict what will be the next one. For example, it's like your input gonna be your input is like the best thing about AI is its ability to, then they're probably gonna predict, there's a high probability, 4% probability to learn. Next one is to predict, something like that. Of course, ChatGPT, they have a random, scheme, inside. They're gonna, every time they'll probably randomly pick from the top signals and, et cetera, et cetera. That give you, a new sentence. So what is the advantage of ChatGPT? There's a, in my opinion, the advantage of ChatGPT is like, no one can read that many, texts, as a person. There's no way you can do that. It's trained by a huge amount of text, all the text available on the internet. And number two is like, GPT really lower the threshold bar for individual to take advantage of AI technique. You don't have to do coding. You don't have to do no Python. You don't have to, write a program. You don't have to, install different package and fine tune that package to make it running. So this is great. And the third one is like, more and more studies suggest, GPT can really give you reasonable good performance when you're just using zero shot. Zero shot basically means you can directly ask a question. Okay, just ask. Most time they give you the, reasonable good performance. All right, so just quickly show a study, how we use the GPT. This is one we use for drug repurposing. So the idea basically like what I said, we don't want to generate our hypothesis using the EHR data because we want to keep that for validation. So we want the hypothesis coming from some other place. So in that case, it's ask my student to say, Chao and Monica to say, can you give us a list? Ask a ChatGPT to give us the drug candidate for potentially can be used for Alzheimer's disease. What we did is this. We ask a ChatGPT multiple times, say, generate 20 candidates for Alzheimer's disease. And then we summarize based on the frequency, we pick the top 10 most frequently mentioned drug candidates. We validate that using EHR data from Vanderbilt and all of us data. Of course, we limit the validation within, over 65 years old, at least 10 years of medical records observation window. We match case and control by age, gender, race, and their primary indications as well. And here are the results. You can see, metformin is we can replicate metformin, we can replicate losartan, and we can, see simvastatin is also interesting finding. And we dig more, and actually, when we search for the clinical trials, and we can see that metformin and losartan right now they are under, trial 3 for Alzheimer's disease prevention or reduce the risk. And I guess the metformin information, the results going to be released 2025. Losartan going to be released in 2028. Okay. Final 1 or 2 slides. EHR is what we're talking about here is just the surface data of the EHR. There's some more and more EHR data going to be available for us. Eventually, I'm always saying that after 10 or 20 years, we may have some patients' whole life EHR data available for us to do. And of course, there's some information is about, we haven't talked about the imaging data as well. We haven't talked about some test data like a pill tone test data and image data. And remember, in the future, smart device information going to be part of the EHR as well. Maybe social media information going to be there as well. Okay, so there's lots of data in the future we can leverage. Going to be linked to EHR, and keep saying that a better phenotyping going to be a key part to leverage the power of the EHR. In the future, I guess that a better phenotyping will enable us to support lots of I guess more and more basic science researchers to help them validate their findings using real world data. And we probably got to do more and more large observation data with a greater power, statistical power, to answer some important questions that I think some questions may not be answered through trials. For example, like the study story I just showed you. And additionally, I guess eventually the EHR data gonna play a more and more important role at all level of scientific evidence generations. That gonna help us drive, deliver better, biomedical research, and eventually accelerate us for the precision medicine. I'm gonna stop here. Sorry, I only have left you probably 4 or 5 minutes for questions.
No, that was great, Wei-Qi. Thank you so much for that very comprehensive overview. So before we get to questions, if anybody attending has a question, feel free to post it in the chat or in the Q&A. And then either I'll ask Wei-Qi or I can unmute you so you can ask. While we're waiting for questions, I do have a couple announcements. This recording will be made available next week on the MPRINT YouTube channel, so you can spread the word to your colleagues who've missed this. The MPRINT webinar series will take a little break for the holidays and then is returning on January 16th for our next talk. And just to put out a general reminder that the MPRINT annual meeting will be here in Nashville, Tennessee, May 14th through 16th. And registration for that information about registering will be made available in the new year on the website imprint.org. So let me see. It looks like there was yes, there's the link to the MPRINT YouTube channel is posted in the chat for everybody there. While we're waiting for questions, Wei-Qi, I had a question about one of the last studies you showed, where you basically asked ChatGPT to come up with your hypothesis for drugs for Alzheimer's repurposing. And so you mentioned that some of the top hits, it turned out there were some ongoing trials. So obviously I can figure out where ChatGPT got that information. What about the other ones? Were they, do you have any idea sort of what the source information was, or did they end up not being very good hits, or tell me more about that?
That's the part that I really like ChatGPT. Initially I thought that probably now, lots of, unreleased medications. But when I got the list from my student, it's very reasonable. There's no cancer drugs there. And it's interesting to me is SSRI medications, they were not there. I guess somehow they potentially already read literature, so there's a safety concerns about those medications. Therefore, they stopped doing that. And the medication they give me just a hypertension medication, lipid medications, and antidiabetic medications. So that makes sense to me. So I guess, of course, different larger model may have a slightly different performance. There's no way we know how they train them. It depends on how they train the model and what the data they use, right? So I guess, in my opinion, there's, to me, I guess, I know your concern is about, if we already got the knowledge, then what ChatGPT can give us. Is that?
Well, no, I mean, actually, I was very impressed. I mean, like I said, the knowledge is out there, but a human can't scour the entire internet to get everything. So
Exactly.
I just wanted to hear what your thoughts were on the rest of it. It sounds like you were also impressed with really all of their hits. So
I'm
that's great.
impressed by the list. Like you said, I think, eventually what we want to do is that, I want to take the power of ChatGPT and guide the ChatGPT to give us to say, we haven't read that, but you got evidence from that. Okay, then we build up on that to say, if that evidence give us a potentially chemical compound linked to a medication, that medication linked to treatment. If we can get GPT to give us, the linkage, that would be great.
Great. All right. And there's a question from Emmeline Raven. She said, this is pretty minor, but on the slide where you compare phenotype markers for type 1 and type 2 diabetes, what did the numbers in the parentheses next to each marker represent?
Yeah, that's the FEMAP one. So the idea basically is that we, given a disease phenotype, we have a list of top related medical concepts from other biomedical terminologies. And so the number basically is you can consider that as a weight. So the higher the number, the more connection between that concept to the disease phenotype. So when you add the weight together, that will give us, an overall number for that particular individual to see how much confidence you are about that patient with a particular phenotype.
Excellent. All right. And I don't see any other questions in either the chat or the Q&A, and it's after 2:00 Central Time now. So we will go ahead and conclude. And again, remember everybody, recording is available on the MPRINT YouTube channel, and we will be coming back January 16th for the next webinar. So thank you, everyone.